Hi guys, let's see if we can finish off the chapter in the next 15 minutes. Let's start with section 4, plants fix carbon dioxide, a cyclical series of reactions that utilize atmospheric carbon dioxide to produce carbohydrates. It is known as C3 photosynthesis, and it involves 3 stages: carbon dioxide fixation, carbon dioxide reduction, and RUBP regeneration. Plants as carbon dioxide fixers. CO2, carbon dioxide, is attached to a 5-carbon RUBP by the enzyme RUBP carboxylase and results in a 6-carbon molecule, which is then split into 2 3-carbon molecules called 3PG, and this reaction is accelerated by RUBP carboxylase, also known as Rubisco. Carbon dioxide is now fixed because it is part of a carbohydrate. Here we have the Calvin cycle, which is the cyclic part of photosynthesis. All right, so we start with carbon dioxide fixation, and then we reduce it, and then we make some more RUBP so that it can continue the cycle. All right, you'll notice that we get 8 ATP molecules here, some NADPH, and 3 more ATP over here on the regeneration of RUBP side. Plants fix carbon dioxide. 3PG is reduced to BPG. BPG is then reduced to G3P. Electrons and energy are required for this stage. This stage utilizes the NADPH and some of the ATP that is made in the light reactions. G3P is reduced and chemically able to store as much energy and form larger organic molecules such as glucose. So here we have the reduction of carbon dioxide. So we have ATP that is being broken down using some of the energy to take 3PG and make BPG, which then uses some of the energy in NADPH to make G3P. As 3PG becomes G3P, ATP becomes ADP plus P, so it's breaking down the ATP, and NADPH becomes NADP. So basically it's taking the energy from the light reaction and making this G3P molecule. The regeneration of RUBP. RUBP used in carbon dioxide fixation must be replaced. Every 3 turns of the Calvin cycle, 5 G3P, a 3-carbon molecule, are used to remake 3 of the RUBP molecules, which is a 5-carbon molecule. So here we have the regeneration of RUBP. We start with 5 G3Ps. We use 3 molecules of ATP, and we regenerate 3 molecules of RUBP. As 5 molecules of G3P become 3 molecules of RUBP, 3 molecules of ATP become 3 molecules of ADP plus P. So basically, the ATP is used to make the RUBP. Plants fix carbon dioxide. The importance of the Calvin cycle. G3P can be converted to many other molecules. The hydrocarbon skeleton of G3P can form fatty acids and glycerol to make plant oils. It can make glucose phosphate or simple sugars that organisms use for quick energy. Fructose, which is a glucose molecule stuck to a sucrose molecule. Starch or cellulose, starch for long-term energy storage, or cellulose for building materials, or even amino acids to make proteins. Here we have a tree, and it shows some of the fates of G3P: glucose phosphate, fatty acid synthesis, amino acids. The glucose phosphate can be used to make fructose phosphate. It can be stored in starch, in roots or seeds, or be transformed into cellulose to form trunks, roots, and branches. Here we have section 5, other types of photosynthesis. The majority of plants carry out C3 photosynthesis. These use RUBP carboxylase to fix CO2 into RUBP molecules in mesophyll cells. However, in hot, dry climates, stomata must close to avoid wilting because they would just lose too much water. So CO2 decreases and O2 increases. So when those stomata are closed, carbon dioxide can't get in and oxygen can't get out. So the oxygen starts combining with the RUBP, leading to the production of CO2, which is called photorespiration. C4 plants solve the problem of photorespiration. They fix CO2 into PEP or PEP, which is a C3 molecule. The result is oxaloacetate, I said that wrong, oxaloacetate, a C4 molecule. And in hot, dry climates, C4 plants avoid photorespiration. The net productivity is 2 to 3 times greater than C3 plants. In cool, moist environments, C4 plants can't compete with C3 plants, though, because they are adapted to survive in hot, dry climates. So here we can see the chloroplast distribution in C4 versus C3 plants. So here we have a C3 plant. All right, you'll notice that it is less ordered than the C4. All right, and we have fewer mesophyll cells. All right, these are organized in the C4 plant much more tightly around the plant vein. That is so that they have better access to the water. CO2 fixation in C3 and C4 plants. So here we see the Calvin cycle in a wildflower. Other types of photosynthesis, CAM or CAM photosynthesis. I'm probably going to pronounce this wrong, guys. I'm having trouble pronouncing words today. Chrysaliacine acid metabolism. I know I butchered that. Sorry, guys. CAM plants partition carbon fixation by time. During the night, they fix carbon dioxide to form C4 molecules, which are then stored in large vacuoles. During the daytime, when the sun is shining, NADPH and ATP are available, but the stomata are closed for water conservation. So the C4 molecules release CO2 to the Calvin cycle. Here we have some CAM plants. Pineapples are an example of a CAM plant that divide the parts of photosynthesis by time. They do part at night and part during the daytime. Photosynthesis and adaptation to the environment. The different methods of photosynthesis each have their advantages and disadvantages, and where you find them depends upon the climate. C4 plants are most adapted to high light intensities, high temperatures, and limited rainfall, so desert areas. C3 plants are better adapted to cold and damp conditions, and CAM plants are better adapted to extreme aridity. CAM occurs in 23 families of flowering plants, and they are also found among non-flowering plants. These are generally your succulent plants, so like cacti and those sorts of plants. And that is it. So if you have any questions, let me know, guys.